Hello students, how are you? This is 11 biology. The subject is to study the effect of quality of light on the rate of photosynthesis. Okay. So the requirement for this, I made a setup for this practical. See here one beaker. I have placed one funnel, one test tube and hydrilla plant here. This is table land and this is cellophane paper. We have to change the cellophane papers red and blue after this. Okay. So count the bubbles in 1 hour and we can get the reading in this setup. Okay. So the requirement for this setup is beaker, funnel, test tube, distilled water, KHCO3 solution, potassium bicarbonate. This potassium bicarbonate is added to the beaker to get dissolved CO2 for photosynthesis process. Okay. Due to KHCO3, this hydrilla plant get dissolved CO2. Okay. So place cellophane paper here. I have taken cellophane paper on the electric bulb. Okay. Take one electric lamp and take 3 types of cellophane papers. Blue cellophane paper, green cellophane paper and red cellophane paper. These are the requirements for this setup. Okay. So in this practical, the principle is absorption spectrum due to blue, green and red light. Okay. In procedure, we take 4 setups. One is for sunlight and other 3 are for red, blue and green. Okay. So in each beaker that is containing the hydrilla plant, add in that beaker potassium bicarbonate to enhance the photosynthesis process. Okay. It create dissolved CO2 to the hydrilla plant. Then allow this setup for the exposure of light of red, blue and green. So take the cellophane papers, create 3 types of monochromatic lights and through this the light is passed towards the hydrilla plant and we can get the oxygen bubbles. Okay. Here the oxygen bubbles are collecting in the test tube. How we calculate the readings? Record the evolution of oxygen bubble in the test tube as a result of photosynthesis and take the reading per 1 minute. Okay, and take the average of that. Observation. What we observe in this, the individual quality of light, the photosynthesis rate varies and higher photosynthesis rate is seen in red monochromatic light. Inference rate of photosynthesis is maximum in red light. There are 13 oxygen bubbles in red light. Then we get more reading in blue light and then we get in green light. The least readings are seen here in green light. Okay. So these are different wavelength of lights which are affecting to the photosynthesis process. Red light is of 600 to 700 nanometer, and it is efficient wavelength for photosynthesis and growth of plant. Height is increases in this red light. Okay. For blue light, the wavelength is 400 to 500 nm. This light regulates the plant's activities, that is the activity like stomata opening, then thickness of plant, and other some morphological activities of plant. So blue light is helping in morphology of plant. Okay. For green monochromatic light, the wavelength is 500 to 600 nm. It get less absorbed by the pigment, and it is crucial for penetrating leaf canopy. So green light is less useful for the plants, and there is the combination effect in photosynthesis when we use three types of monochromatic light. The most efficient photosynthetic process is achieved by using these three combination of light: red, blue and green. They are of different wavelength. It is supplemented by green and far red monochromatic light of different wavelength, and it maximizes the plant light height. Okay. So this is about the quality of light on the photosynthesis. What are the effects of qualities? So students, if you like the video, you check my other published videos and subscribe our channel and write your comment in comment box.